And now we have 3 brief presentations. The first by Dr. Georgina Butler, a research associate in Dr. Chris Overall's lab here at the Center for Blood Research. I'm going to hand it over to George.
Okay, so I don't have long, so this is a need-to-know basis. So 3CL-Pro is a protease from SARS-CoV-2. So when SARS-CoV-2 gets into the cell, its RNA is translated into 2 viral polyproteins. I don't have a pointer, so I'm sorry about that. And these polyproteins encode 16 non-structural proteins, or NSPs. 3CL-Pro is NSP5, and it's a protease. And there's also another protease called PL-Pro, NSP3. And these proteases are essential because they cut the viral polyproteins into constituent active functional viral proteins. And what you need to know is that 3CL-Pro is a cysteine protease, and the key catalytic residue is cysteine 145. This structure's from CBR Stranakalab. And 3CL-Pro likes to cut in the bottom right at that cleavage site, LQ or H, and then a G, A, or an S. So how does this tiny virus overcome the mighty human? We know that 3CL-Pro is important because a 3CL-Pro inhibitor is one of the components of Paxlovid. So the way we wanted to ask whether 3CL-Pro, how that interacts with our own proteins, what pathways is it involved in, and what proteins does it cut. So we used inactive catalytic domain capture. So you can see the construct there in the middle in red. We expressed an inactive protease, where the catalytic cysteine is mutated to alanine, and our construct had a couple of tags so that the protease can be isolated, and an additional mutation to mutate out an autocatalytic cleavage site. So the methodology is really to make a protein extract from cells of interest, do a brief incubation with the inactive protease, which can then capture proteins, interactors, and substrates, which are not cleaved and therefore not released, so they're trapped, and also do a parallel capture without any protease to rule out non-specific interactions from the anti-MYC or anti-flag magnetic beads. And then we did isotopic labeling to be able to differentiate between specific interactors and non-specific interactors. So what we ended up with was 259 proteins that were enriched in the 3CL-Pro pullout. And we found that a large number of these were cytoskeletal proteins, or proteins involved in cytoskeletal organization or in cell projections. So next we looked at infected human lung epithelial cell line to see if there were any changes in cell shape or any projections. And you can see in the photograph in the middle at the bottom that indeed in the infected cells, SARS-CoV-2 infected cells, we saw lots of really thin anuclear bridges connecting distant cells. And we actually saw a few of them in the mock infected cells. And in the graph on the right, you can see that in the infected cells, we got an increase in these, what we are now defining as tunneling nanotubes, or TNTs, with viral infection. And if we used a 3CL-Pro inhibitor, we reduce the number of tunneling nanotubes. So tunneling nanotubes are known to be produced by cells, and they enable cells to transfer materials, including organelles as big as mitochondria. And other viruses actually do induce tunneling nanotubes to promote infection because it's then much more efficient and immune protected. And you can see on those stainings that we did a time course of infection with SARS-CoV-2, and we found there was nucleocapsid protein inside these tunneling nanotubes. And another publication came out showing that SARS-CoV-2 can be transmitted through tunneling nanotubes between cells that have the ACE2 receptor and cells that don't, for instance, neuronal cells. So how might 3CL-Pro be involved in forming tunneling nanotubes? So what we did was we took the 259 proteins and scanned through the protein sequences for canonical cleavage sites, which is shown top left. And those 13 proteins there were confirmed as substrates of 3CL-Pro that are involved in the cytoskeleton. So to confirm that they were substrates, we used a variety of assays, which are shown here, and you can come talk to me after if you have any questions. So we confirmed basically that they were substrates in vitro, and then we looked in lysates from infected cells, where we saw that these proteins were cleaved at 24 and 48 hours post-infection. And then for 5 of the substrates, we looked in normal lung and post-mortem COVID lung, and saw that those proteins were decreased in the COVID lung. So many of these proteins, when we did immunolocalization, were found at the edges of the cells and the edges of the cell colonies where they co-localize with nucleocapsid, viral nucleocapsid protein. And when I was looking to see what these substrates interacted with, to work out what cleavage might be doing, I noticed that a lot of those proteins in the interactome that were cleaved, shown in dark green, that were found in another methodology that we have called tails, in light green, and also other interactors, interacted with at least 3 proteins that are shown in yellow in that middle diagram, that form scaffolds in the adherens junction. I'm just showing cadherin 1 here. So all of those proteins that interact with 3CL-Pro, or are cut by 3CL-Pro, also interact with that scaffolding protein. And then if you look at the top right, what we think is happening is that the proteins shown in green up there that are in these big complexes are being cleaved by 3CL-Pro, and then that allows rearrangement of the cytoskeleton to form these tunneling nanotubes. And you'll notice that, I don't know if you can see, that there's also transcription factors that are sequestered in those complexes in what's known as the zonular signalosome. So we also think that cleavage by 3CL-Pro releases these transcription factors, and it's known that in SARS-CoV-2 infection there are big changes in gene expression. And if you look in the bottom left, you can see that one of those in the hipposignaling pathway, YAP1, is cytoplasmic. These are Vero6 cells, so they look different. It's cytoplasmic in mock-infected cells, but when you have infection, it does translocate to the nucleus. So we think that 3CL-Pro cleavage might be a mechanism for allowing that. And we also know that YAP binds to promoters of some of those genes, the proteins that are cut in infection, so that could replenish the cytoskeletal components and also provide building blocks for the growing tunneling nanotubes. That's all I've got time for. So if you'd like to know more, I'm at poster number 11. That there.
Thank you. That's great work. I was wondering, can we make antibodies against the protease after an infection? I'm sure we can't after vaccination. And would that be effective as a target?
So the protease, it is a target, which is why it's part of the I don't know how to say it, the nirmatrelvir, whatever it is, it's in Paxlovid. And other work in our lab has shown that the 3CL-Pro does get out of the cell. And also there's a lot of cell death, so it probably is outside of the cell. Do you mean us making antibodies, or do you mean are there antibodies that we could use to target?
After an infection, do we make antibodies against it? Because presumably that wouldn't mutate a lot if...
yeah, I don't think anybody's known. I think there are mutations in the 3CL-Pro.
Oh, there are?
Yeah, but I don't think they're that dramatic. But I'm pretty sure that nobody's looked for antibodies because it's not actually well known that it gets out of the cell.
I only ask because I have a cohort of patients who have been infected versus exclusively infected versus exclusively vaccinated. And I'm wondering if those antibodies would be neutralizing if you make it against the protease.
And there were a couple of commercial antibodies against 3CL-Pro, but they were not very good. So we made an in-house one. So we do have an in-house antibody, but maybe if it's...
Well, it'd be polyclonal, so it would be good. I'm guessing it would be good. Anyways, worth looking at. Thank you.